Hello everyone, I am going to deliver a lecture on the topic antibodies, its structure and function. So an antibody is a protein produced by immune system that is capable of binding with high affinity to an antigen. So these antigens are typically other proteins but may be a carbohydrate or a small molecules or even a nucleotides. The specific region of an antigen that an antibody recognizes and binds is called as epitope or antigenic determinants. So serum containing antigen specific antibodies is called as antiserum. So there are 5 classes of immunoglobulins are there, including IgM, IgG, IgA, IgD and IgE. Now we will see the structure of an antibody. So antibodies are made up of identical to light chains, which is made up of kappa or lambda, and identical to heavy chains, which are made up of mu g, gamma, alpha, delta and epsilon. So each light chain bound to a heavy chain by a disulfide bond to form a heterodimer. So 2 identical heavy chains and 2 identical light chains combine to each other to form a 4-chain antibody structure, a dimer of dimers by non-covalent interactions and disulfide bridges. So the exact number and position of these interchain disulfide bonds differ among antibody classes and their subclasses. So the first 100 amino acid terminal of both heavy chain and light chain vary among antibodies of different specificity, and these regions are called as variable regions or V regions. So this is referred as variable regions in light chain and variable regions in heavy chain. So differences among antibodies fall within the areas of these variable regions are called as complementary determining regions, and it is this CDR on both heavy chain and light chain that constitute antigen binding site for the antibody molecule, and this is called as Fab region. It is nothing but fragment antigen binding region. So this will be there on both heavy chain as well as light chain. So the regions of relatively constant sequence beyond the variable regions are called as constant regions, which is called as constant regions on heavy chain and constant regions on light chain. So antibodies are glycoproteins with a few exceptions. Glycoproteins are nothing but this is a protein which is also consisting of protein. The sites of attachment of this carbohydrate are restricted to the constant region. That means the carbohydrate will bind only to the constant region of the antibody molecule. The other fragments had no antigen binding activity at all because it was found to crystallize during the cold storage. It was called the FC fragment. It is nothing but fragment crystallizable region. So the function of this fragment is nothing but which will bind on the FC receptors on the phagocytic cell so that they will be inducing opsonization. So the another region is called as hinge region that will be there on the antibody structure, is rich in proline residues and it is very flexible. So because of the flexibility, 2 Fab arms can assume various angles to each other when an antigen is bound. So these are the different structure which is there on the antibody structure. So the one end of the antibody is called as amino terminal and other end of the antibody is called as carboxy terminal. So now we will see the different classes of antibodies. So each class is distinguished by unique amino acid sequences in heavy chain constant region. So confer class specific structural and functional properties. The very first class of antibody is immunoglobulin G, or it is also called as IgG. So IgG, it is the most abundant class in serum which is constitute about 80% of total serum immunoglobulin. The IgG molecule consisting of 2 heavy gamma chain and 2 kappa light chain. So there are 4 human IgG subclasses are there which is differentiated by differences in the gamma chain sequences and numbered according to their decreasing average serum concentrations. So the first one is IgG1 and IgG2, IgG3 and IgG4. The structural characteristics that distinguish these subclasses from one another are the size of the hinge region and the number and position of the interchain disulfide bond between the heavy chain. So amino acid differences between subclasses of IgG that will affect the biological activity of the molecule, where the IgG1, IgG IgG3 and IgG4 readily cross the placenta, so they play an important role in protecting the developing fetus. So IgG3 is the most effective complement activator, followed by IgG1, IgG2 is less efficient, and IgG4 is not able to activate the complement at all. So IgG1 and IgG3 bind with a high affinity to Fc receptors on phagocytic cells and thus mediate the opsonization, where IgG4 has an intermediate affinity for Fc receptors and IgG2 has extremely low affinity towards the Fc receptors present on the phagocytic cells. The next class is IgG-M, immunoglobulin M. IgG-M accounts for 5 to 10% of total serum immunoglobulin with an average serum concentration of 1.5 mg per ml. So, the monomeric IgM with a molecular weight 1,80,000, which will be expressed on the membrane bound antibody on B cells, but IgM secreted by the plasma cells as a pentamer in which 5 monomer units are held together by disulfide bond that link their carboxy terminal heavy chain domains and monomers of C3 and C3 domains. The 5 monomer subunits are arranged with their Fc regions in the center of the pentamer and the 10 antigen binding sites on the periphery of the molecule. Each pentamer contains an additional Fc linked polypeptide called as J chain, which is disulfide bonded to the carboxyl terminal cysteine residue. The J chain appears to be required for the polymerization of the monomer to form pentameric IgM. So, IgM is the first immunoglobulin class produced in primary response to an antigen, also the first immunoglobulin to be synthesized by the neonate. Because of its pentameric structure with the 10 antigenic binding sites, IgM molecule can bind 10 small haptane molecule. Because of the steric hindrance, it can only possible to bind the 5 haptane molecules. So, IgM is also more efficient than IgG at activating the complement.